Well, good evening, ladies and gentlemen, and welcome to this BOFIN webinar on genomics. Apologies for the slightly late start. My name's Tom Allen Stevens. I lead the British On-Farm Innovation Network, and this is a very new area of plant science that we will be exploring. So I'm a little bit excited about what we'll find out. Now, I ought to clarify here that what we mean by genomics. Unlike genetic modification or gene editing technique known as CRISPR, genomics is a field of science that uses genomic information to study how genes affect plant health and biological processes. It doesn't alter DNA, but analyzes genetic data to unlock insights. Now, I'll leave it to the scientists to explain that in more detail later on. But first, I just want to bring you up to date with what else is coming up at BOFIN. Now, we've had a busy couple of months across the 4 major DEFRA-funded projects that we're running. The Soya to Sustainability Conference we put on last month in Peterborough was a huge success, bringing together the whole of the supply chain to recognise the value of homegrown pulses over imported soya. Now, do check out ncsproject.co.uk for a roundup of the conference. If you're one of the 73 farmers that were paying to be part of our trials, first of all, thank you. We've just about had all the data back from the slug sloughs. Processing is now underway. Resource packs are on their way out to root rangers, undertaking intensive soil sampling this spring. We'll also put together the shovelomics guidance. We've also put that together and look forward to the first few assessments coming through on what roots look like this spring. And we'll be sending out PES sensors shortly, so you can do some analysis directly on your own fields. All the outcomes of this analysis will go first on the soil circle, so anyone can join this community to take part in the project. So do keep an eye out for more. Now, hot off the press, announcements were made on the DEFRA funding for trials and precision bred crops at the NFU conference this week by Steve Reed. So BOFIN was there, and to summarise briefly, the secondary legislation for precision bred crops has now been put in front of Parliament. So this is the regulatory process that will determine what is a PBO or a precision bred organism, and then how it can be incorporated into our food. If passed by Parliament, then there will then be 6 months before the first applications can be approved. So this now means that it's unlikely that the PBOs that we're currently multiplying up as part of the Probity project will be fully approved before this autumn. But all 3 can be planted in the spring. So we're still hoping to do it for this harvest at least. What's more, we have 2 tilling lines that don't require the PBO approval, and we'll be looking for growers interested in trialling them this autumn. So look out for that if you'd like to be involved in these precision bred crops. So BOFIN is a key consultation on this process through the Probity project. Both DEFRA and the NFU want to know your thoughts on these new technologies as the legislation passes through Parliament. It's really, really important that you take that you make your thoughts known. And the Sequence Circle is the best platform to do this. So this is our new circle on the Farming Forum. So please take that opportunity, take the Probity Pledge and spend at least an hour finding out about gene editing, and then bring your thoughts to the Sequence Circle and help shape the crops of the future. Now, there's mixed news on farm innovation. Government has committed funding to adopt to the Farming Innovation Programme, also to the Farm Equipment Technology Fund, which is good. And that's good news that they've lifted the freeze on funding and it signals progress. So you can register your interest in Adopt through the BOFIN website. So do take a look at that. But the amount of funding pledged, I'm afraid, has fallen way short of what's needed. In fact, it's about 10% of what the NFU was campaigning for. But nonetheless, that makes it all the more important that we get going as soon as we can. And if you're looking for funding for on-farm trials, this is a really great opportunity. So also this year, BOFIN is exhibiting at a number of key events this year. We're an event partner for cereals, which is really great news. And we'll also have a stand at Groundswell, and we're hosting panel discussions at both of these events on precision bred crops. So Do please come along and take part in those events. We'll also be hosting the first BOFIN Inspiration Days in May on farms, on our host farms, where trials will be taking place. It's a chance to come along, network, and help shape those trials. And all of our trialist farmers will be encouraged to hold an Innovation in Action days this year. We'll also be supporting them to host an event or farm walk for their local farmers. And also look out for the trials results. Our farmers did a sterling job amassing a plethora of readings, observations. Sorry, my computer's doing an update on Zoom and it's just completely going bonkers. Our farmers have done a fantastic job amassing all the data this year. There'll be a plethora of trials results to come out later on as well. So you'll hear about all those results and the conclusions as the scientists work through the data. So, well, that's about enough from me. I'm going to be handing over to John Hayward from Unium to introduce this really exciting topic, and then take it on from here. So, John, are you there? Would you like to make yourself known?
Yeah, so thanks, Tom. Yeah, I'm John Hayward of Unium Bioscience, and I guess I've been in this industry of biostimulants and biologicals now for over the last 25 years. And have constantly tried to lose the snake oil image of the industry. Some might say it's hopefully not to lose it from me personally. I'd hope I never collected that image, but you never know. We've done that by using peer-reviewed science, lots of independent data to create reliable solutions. But there's always that cynical challenge that we face year on year and that we have to overcome. But now through our tremendous partnership with HELM, and in particular linking with Tim Eyrich, who you'll hear later, and Layne Ellen, who we're really grateful for the work she's done, particularly for Tim and ourselves. We can take this to the next level with genomic studies. That links plant physiology, plant genetics, nutrition, and the biostimulant biological platforms to create solutions for farmers and growers. One or two comments have been made as to why Unium getting involved in genetics, etc. And as Tom covered earlier, we're not getting involved in genetics as some might see. And the best analogy that we can have really is we're going to learn how to read the book without changing the text. GMO CRISPR may change the text, and they look to change the words. What we're wanting to do is read the book and read it at a more detailed level. And then another analogy is really it's like having a SAP analysis of the genetic makeup of the plant pre and post any treatment. So we can look at stresses, look at impacts on the plant, and then look to see what technologies we have can mitigate those. So this allows us to build a really detailed mode of action of our technologies and products. We can create quicker solutions to the problems that are arising and changing all the time. And that gives us a more targeted, accurate approach to achieve our objectives, but importantly creates reliable and robust, and now through this technology being a lower cost, affordable solutions for farmers. And it's really interesting because if you think about it, with around 60 to 70% of the crop production losses, they're lost to abiotic stress. So the environmental stresses that we're all facing, compared to biotic stress, diseases, weeds or pests, which only take around 15 to 20%. But in contrast to that, only around 40% of farmers are using products, be they biostimulant biologicals, to mitigate these abiotic stresses that are taking the majority of our lead. So hopefully with the aid of this technology, we can bridge the cynical past with reliable science, creating solutions for the future. So with that little coverall, I'm going to hand over to Ben Taylor-Davies, Regen Ben, as most people will know him by, who's really kindly offered to chair this session, along with the two main scientists and agronomists, Tim and Layne Ellen. So we hope you enjoyed the webinar, and the recording will be available tomorrow. So without further ado, I'll invite Ben to take the chair.
Thank you very much, John. Welcome, everybody. I'm not going to talk for very long because we've got two very exciting people who I had the pleasure of having. I think it was an hour Zoom the other week that seemed to pass in what felt like 5 minutes, and we ran out of time, and it was a real shame. So what we learned and what I found out was just quite phenomenally exciting. I'm the ultimate cynic when it comes to snake oil, and really was anti every part of the snake oil sales until I stumbled across, and John offered me the chance to use Scion many years ago now. And for the first time ever, I actually saw something that had been peer reviewed, had got some very excellent data behind it, and it actually worked. And that really started to change my whole perception of what is essentially biostimulants. Um, and opened up my eyes to the fact that actually if we can help the plant and help the plants naturally to fight off disease, pests, and everything else, abiotic stresses, then in actual fact this could have a huge impact on yields and maintaining everything we're looking for in a crop and a healthy environment. So without further ado, I'd like to introduce first of all Layne Ellen, who I believe is from Kentucky, and yeah, absolutely, who's PhD and just absolutely phenomenal knowledge on how these compounds and nutrition has this massive effect on switching on genes to their natural defense responses. So over to you, Layne.
Thank you so much, Ben. I'm actually from the other UK, which is the University of Kentucky. So sometimes I've been mistaken for being from the UK before, and then it only took about 5 seconds of listening to me to realize that I was from the redneck state of Kentucky. So I'm going to share my slides here. I'm so pleased for the invitation and to get to be here today to talk to you all about how we use genomics and genomics research in understanding biologicals. Let me share here. All right. So, so my company, I should also introduce myself. My name is Layne Ellen Harris. My company is Foresight Agronomics, and we're a research consulting firm that our goal is really to demystify the use of biologicals by looking at how when we apply these products to plants, we can influence how their genes are expressing. And today we're going to talk a little bit about what does that really mean when we say, look what their genes are expressing and why would that matter? And so we're going to walk through some technical work, but also a few examples. But I have to first start with a little geography lesson. I know that you probably did not log in here to learn about Kentucky, but I cannot help myself. I promise it is relevant. So if you're not familiar, Kentucky is a pretty rural state in the southeastern part of the United States. And I am from a tiny little county over here called Lyon County in the western part of the state. And I want to point out that there's a couple of these really big lakes here. So just for scale, these two lakes here are the largest man-made lakes east of the Mississippi in the United States. They are absolutely massive. So this is what it looks like now. These lakes bring in a lot of tourists, a lot of fishing, and we're going to have a little bit of a redneck fishing analogy today. I hope you don't mind. So if you're not familiar with the geography of where I'm from, Kentucky is known for these kind of lush rolling hills. We're called the Bluegrass State. And there's some interesting history about the part of Kentucky where I'm from and why these lakes are relevant to what we're even talking about today. So just to familiarize yourself, though, this is kind of what the terrain looks like in the part of the state where I'm from. Now it looks like this. And so back in the early 30s, the Tennessee Valley Authority actually made these two lakes by building dams on a couple of rivers there. And there were some unfortunate things with lots of families being displaced with these millions of acres of flooding, essentially. But when they created these lakes, they also, they really covered what is still under the water, which is all these hills and valleys and what would have been cities and towns and railroad tracks and all these things. And so the reason I bring this up is my dad is a pretty avid fisherman. His dad remembered what the land looked like before the lakes were built. And he actually taught him where all the different geographies would have been under the water. And so my dad, being the exceptionally smart man that he is, used this information to optimize his fishing adventures, right? So he knew where the crappie and the bass and all these fish that he's trying to catch are going to be, because he understands what's under the surface of this water. Now, I have to tell you a funny story about another reason this was particularly relevant. So me and my dad were out fishing one day, and I want to paint the picture for you. So again, we're in Kentucky. We've got some, stereotypes that I won't say are totally incorrect either, but me and my dad are out fishing in his John boat, which, is kind of like this. It's a pretty small little boat, and we're out catfishing. And my dad looks out into the middle of this lake. And again, these are huge, huge lakes. And we see this boat coming along that looks something like this. I mean, just your luxury yacht, because being these big lakes, we get a lot of out-of-town people that come and vacation there. But what was interesting about this particular time is that my dad just starts laughing, and I don't really know what's going on. I just see this huge boat coming down the middle of the lake, and he is just cracking up. And not long after that, we see this huge yacht suddenly stop in the middle of the water, which, if you've ever been around boats or watched boats do anything, they don't typically suddenly do anything, right? So this boat suddenly stops, and we hear some guys yelling and cussing, and we hear a splash, and then I see this man walk around around the outside of the boat in the middle of this lake. And what had happened was they had actually gotten themselves stuck. They did not know what was under the surface. There's still these rolling hills under the surface of this lake, and this yacht actually got stuck in about 3 feet of water. And it was hilarious to watch them get towed out. And me and my dad sat there and ate our bologna sandwiches and watched this multi-million-dollar yacht get pulled out because they did not know what was going under the surface of this water. So why do I even bring this up? Whenever we look at how products are affecting the expression of genes in a plant, we're able to look under the surface. Like John said, this is kind of a SAP analysis on steroids. We're able to look at real time how a plant or how a crop is kind of perceiving this thing that we're doing to it. And gene expression is one of the ways that we do that. So whenever we think about biostimulants, we generally think of them in terms of, we have this certain product, it has a given application, and that leads to some kind of expected results, right? But there's a huge question mark on how does that get there, right? How does this product applied at this rate and these crops at this time actually get us to these expected results? And that's really where I use gene expression to come in. So now we're going to talk about gene expression. Hopefully this won't put you to sleep. Maybe you've got some coffee with you, but we're going to go very basic and explain what this is. So here's a plant cell, and in the nucleus of the cell, or right here in the middle, is where all of the genetic material, and there's also some in mitochondria and chloroplasts, but that's beside the point. The genetic material in the cell is in the nucleus of the cell. This is where the DNA is. Whenever we say that a gene is expressed, what it's doing is it's accessing regions of DNA and reading it like a code, reading a message. So this is called the central dogma of biology, or molecular biology, which is essentially how information is transferred in a cell from DNA to RNA to a protein. And so whenever we, let's talk about what that is, and we don't have to go into too far detail. There are lots and lots of nuances and regulatory systems that cells use to maintain this and protect this pathway. But essentially, whenever a gene is expressed, so like let's say that a plant is getting stressed and it needs to access some of its abilities to deal with, let's say, drought stress, it will go and find regions of that DNA that has genes that code for proteins that give that plant special abilities to deal with stress. Maybe it helps it not lose water. Maybe it helps it protect some of the enzymatic functions of things in the cells. And so that gene or sequence is located in the DNA, but the DNA is stuck in the nucleus of that cell. And so what, in an amazingly efficient and elegant way, that DNA unwinds. So it's 2 strands, it unwinds, and a copy of that DNA is made. And that copy is like a little mobile version of that gene. And that little tiny fragment copy can leave the nucleus of the cell and go to where it needs to go to be translated into the enzyme or the protein that this gene is actually the code for. So an analogy for this might be, if you imagine for a minute that you had a library in your house that was full of recipe books, and you want to make a delicious chocolate cake, and you need the recipe for this chocolate cake. Imagine that in your library, all the books are stuck in there, but you can go to your library, find the recipe of the chocolate cake that you want to make a copy of it, and then that copy you can take to the kitchen where your flour and your butter and your sugar and all of your ingredients are to actually make the cake. In gene expression, if you stick with our little analogy, imagine every additional copy of this recipe that you made, you wound up with another cake. We can kind of measure through sequencing and through things like PCR, we can actually measure how many copies of these genes are being expressed. And so, and we can identify what genes are being expressed. And so in any typical crop plant, there are tens of thousands of genes available in this library of DNA in the nucleus of the cell. And so what we can do through sequencing technology and through understanding this central dogma of how information is moving from the plant is accessing information from the DNA and expressing that, if we can identify this little in-between spot right here called mRNA or messenger RNA, then we can actually see what genes a plant is turning on or turning down in response to stress, in response to the developmental stage that it's in, or in response to biological products that we put on it. And so we really utilize this. I won't go into this, but this is an example. In drought stress, we know that there are certain triggers that will happen when a plant experiences drought stress that trigger lots of pathways in the plant, biochemical pathways, but also genetic pathways. And it makes a lot of sense because it's needing to access its ability to make all these things to deal with this drought stress. And so if we think about the result of this, of all these genes expressing, is that now it has the ability to resist some of this drought stress or use its water more efficiently, or maybe continue to grow despite the stress. And by identifying those genes, we can see, can our product basically elicit some of these same responses in a plant, make it think that it's being stressed, but actually cause it to be more resistant to stress down the road, which I know that it's, in my experience and from the testimonies that I hear from people, very often we will see biologicals have some positive effects. And we may think, yeah, I mean, it seemed to help deal with stress more. It seemed to help with yield, or it seemed to help with disease management, but we just can't really figure out why. Or we may have a program that we apply it a certain way one year and we apply it what seems like the same way the next year, and we get two totally different results in the field. And so the idea of this type of approach is to really, really narrow in on what things is our product doing to the plant? How does that plant perceive that thing? And we measure that by the types of genes the plant is turning on or turning down in response to that. Another example of this is in heat stress and cold stress. So I look a lot at abiotic stress, priming and resistance and tolerance and recovery. There's a lot of nuance there, but again, there are these pretty well-established pathways that we know a plant will use to deal with its environment. And so if we kind of overlay that with the responses that we're able to elicit with a product, then we can start to get extremely wonderfully strategic about how we're using this product, given the type of crop that we have, when we're applying it in that crop's life cycle, if there are other stresses going on, or the nutrition that's required to actually fuel these pathways. So even in this pathway here, looking at it, there's things like calcium involved, phosphorus involved, potassium involved. These are really, really important pieces of this signal transduction that the crop has to have enough of in order to do this, which is why I'm sure Tim will talk about the nutrition matters so much to give your crop the best ability to fully express these mechanisms that we can use biologicals to nudge it into. So a specific example of this, so we're back to this idea. We have a biostimulant, we apply it in a certain way. How are we getting these expected results? I want to show some real data with you. This is what this is some of the outputs that you get with a typical project like we would do. So this is actually this is real data from a project that I contracted out myself last year with a biostimulant product that we knew had some positive effects on crops, especially grain crops. It seems to, growers said that it seemed to help with some abiotic stress tolerance, and the plants were a little bit taller. And so we started to look at what the genes are that our product was affecting the expression of in these crops. And so again, we're not adding in new genes. The plant is expressing these things all the time. This is just a way for us to look at what it's doing. It's not going in and changing that. And so anyways, we don't have to go into too much of the data, but this is what's called a volcano plot. And what this shows is all these red dots and green dots are genes that have been statistically significantly affected by our treatment, or in this case, it was a biological product or a biostimulant versus our untreated control of water. And so we can see that our product at this point, and this was at 24 hours after application, there were 351 genes that were significantly upregulated, some of them very significantly at sevenfold upregulation. So it's expressing some of these genes 7 times more than the negative control. And on the flip side, we have genes that are being downregulated, 388 genes that are being suppressed and turned down, which is not necessarily a bad thing. Some pathways in plants are actually, it's kind of like if you were parked on a hill and you took off the brakes on your car, you go forward. Some pathways in plants are kind of like, by stopping the brakes, you actually allow the process to go forward. So we look at both genes that are upregulated as well as downregulated. So now, if we look at, this is a heat map. This is a pretty typical way of demonstrating a transcriptomic analysis results, but just to very quickly orient you to this, each of these columns is one of my samples, and each little tiny row here is a gene, and the color indicates if it's being upregulated or hot, or if it's being downregulated or cool. So green would be a negative downregulation of genes, and red is upregulation. The main thing to look at here is that our treatment sample, these three here, have a whole bunch of genes that are red that are green in the control samples, and vice versa. There's a bunch of genes that are green in our treated samples here that are red in our control. And what that tells us is that this product is significantly affecting the expression of a lot of genes. We're seeing tons of genes being turned on in our products that got the biostimulant that are not being turned on in our product that only got water. So we can establish that there's an effect. Great. So what? What are these genes? What difference does it make if we are doing this, if that doesn't tell us why we're seeing this response? So we won't have to go into all of this unless you want to geek out on biochemistry. I'm happy to do that. But essentially, when we look at some of these genes and how they operate within pathways, which is really, really, really important when you look at gene expression data, it's important to know that while you might affect the expression of a gene, of a single gene, very significantly, there are lots and lots of genes that are involved in these pathways that the plant has to use to make hormones and to make defense molecules and to grow and to make photosynthetic cells and chlorophyll and all these things. There are hundreds of genes involved in all of those processes. And so I find more value from looking at, are we affecting entire pathways? Because truthfully, that kind of information is probably what's going to actually give you explanations as to what you see in a field versus what you might see under certain conditions in a heavily controlled environment that you might see from like publications or more academic kinds of trials. And so When we look at this, this is called an enrichment analysis, and we don't have to go into all of these, obviously, but some of the main things that stand out to me looking at this data here is that we see there's a little ping here on nitrogen metabolism. So maybe our product is actually causing the plant to take up more nitrogen or to use the nitrogen that it already has a little bit more efficiently. We also see things like starch and sucrose metabolism, which is your sugars. That tells me that there is, when we put this product on the plant, it is causing the plant to need to use energy. It is having a response that the plant needs to use energy in order to do. And so that's not necessarily a bad thing, but what that can mean is if we apply too much of the product or we apply it too frequently, we run the risk of actually diverting too much energy away from growth, so that the plant, we're trying to constantly trigger it to do this thing, and we run the risk of actually overdoing it and causing it to put too much energy towards this response. And then there are lots and lots of other responses here. There are things like phenylpropanoid, which tells me that it might be helpful in disease resistance. Much of the plant immune responses that help with disease resistance are products of this phenylpropanoid pathway. And so again, I deep dive, truthfully, I probably spend too much time looking at this data, but it's absolutely fascinating. And so when we think back to some of the expected results from this product, one of the things is plant growth. We saw that it seemed to generally result in some taller plants. Well, when we look at some of the expression on very specific hormone pathways, we see that there is a significant increase in genes involved in the auxin signaling pathway. So if we look at this pathway as shown here, this is our auxin receptor. Auxin is a growth hormone that helps with above ground tissue. We can see that genes that show that the plant is using more auxin, responding more to auxin, are being upregulated and fairly significantly as well. So now that we've gotten this far, it's no longer this big question. We can actually explain why we see that plants are taller, why we see that there may be times that we can actually overdo this product application. Certain plants may be more sensitive based on that starch and sucrose, that energy metabolism that's required to deal with this. We can see that there are certain nutrients that are required in order for the plant to be able to respond and most efficiently get primed, I guess you could say, for these types of stress responses or stress resistance that it might encounter later in the season. So why does this matter? Well, we can answer questions about durability. If we take samples throughout the season, we can see how long does this response last? Do we need to apply this product multiple times, or is one time enough? Or does it really start to not really matter that much after 3 times? You can actually measure this response and see how it tracks over the season. Alongside that, we can also see, are there particular time points in the crop's developmental stages that it's going to make the most sense to utilize this product? Now that we know what it's doing to a plant, we can know, okay, maybe it is working to stimulate defense responses that makes it more resistant to fungal diseases later in the season. Well, I know that based on what I've seen in, let's say, corn genetics, the plant might be a little bit more receptive to that kind of response during, a V4 or V5 early vegetative growth. But as it gets closer to reproduction, it begins to get a little less focused on defense and a little more focused on reproduction. So if we're trying to prime this plant for disease, I want to try to get an application in early. And if I wait too late, I might not get as good of a response. We can see, like I mentioned before, does applying multiple times help? We can see, is this a systemic response? Does it matter if we put this in drip or in furrow or spraying it foliarly? And then finally, like I mentioned before, we can see if there are indications that the plant is getting stressed whenever we put these products on it. And so that can give us some information about there may be too high of a rate. I know biologicals are generally thought to be safer in terms of the risk of phytotoxicity and that kind of thing. But I've certainly seen cases of diminishing returns after a certain amount where you're no longer further eliciting a response. You're either throwing away product or you're beginning to overstress the plant. And so we can actually see that there are limits to certain rates, or that you need a minimum rate, which again, sometimes these products are notorious for, you just put in a pint an acre and it doesn't matter what your carrier volume is. Maybe it does. It might matter if you have 10 gallons per acre, or I'm trying to think about metrics. So apologies for that. I can't immediately do that math in my brain. But if you have the kind of carrier volume that you have with corn, it might be very, very different for apples. And so we can actually see, are there minimum rate requirements that are required in order to elicit these kinds of responses, and elicit them to the point that it actually gives you measurable results in the field. And then finally, we use this approach a lot in research for seeing where we need to go with our future directions. We can use this to, as I've already said, identify modes of action or candidate modes of action. If we want to optimize our formulation of a product, or how this product might really synergistically pair with other products that you're putting out, we can actually really get some good answers to questions around that and get really strategic about what next steps look like. So back to my lake. Why does it matter? Well, number one, if you know what's under the surface, if you know what's going on, you can get incredibly strategic with how you're using these products. You know, it's not a guarantee that if my dad goes out that he's going to catch fish, but he's really increasing his likelihood if he knows where they're probably going to be and when they're probably going to be there. He's going to really increase his likelihood of catching what it is that he's fishing for. And on that same token, you can avoid a lot of risk by knowing what's under the surface. I think about that luxury yacht, and I think in that moment, it did not matter that that thing cost tens of millions of dollars. It was stuck because they didn't know what was under the surface. And so this can be a wonderful tool to not only get really strategic on when you're using products and why you're using certain products, and what you're trying to accomplish in your crop, but also help with some of the risk management of knowing how to navigate some of those underwater hills, we'll say, that that you wouldn't know about if you didn't know what was under the surface. So with that, I'm not sure where I'm at on time. Hopefully I've done good. I don't see a clock. So if you have any questions, I'm sticking around and happy to talk more about this. Or if you ever want to email me directly, please feel free to reach out.
Excellent. Thank you very much for that insightful information. I think without further ado, we'll get straight on to Tim, who's head of agronomy at HELM Agro. Tim, one of the interesting things looking at the website of HELM, I thought it was quite interesting the way you market biostimulants as plant advantage products. I think it's probably the best way of describing these things, really. So, yeah. We're more than interested to listen to what you're going to say about plant advantage products.
Excellent. Thank you, Ben. And thank you for allowing us to talk today. It's been doing this for about 44, 45 years. Is my screen still showing? Got it. Can anybody hear me?
Yes, all loud and clear.
Yeah, I'm trying to make sure I got it back up. Okay. And really in the last 5 years, being able to use this technology has allowed us to do several things. Our biostimulants work more consistently. And so what I'm going to talk about today is how do we take this genomic data and put it into decision-making at the field. Just a little bit about my background. I got a degree in agronomy, plant physiology from the University of Arizona way back in 1983. I was blessed with two really good men in my life early. Dr. Lee Stith was a cotton breeder. He was my advisor, and he said, taught me to never, ever forget the source to sink relationship. It says everything you're going to do in agriculture, Tim, as an agronomist, is going to be based around the source to sink relationship. And that's, that's growing plants. It's making healthy leaves, taking sunlight, turning it into food. This kind of shows us that. What I'm looking at here's nice, healthy corn plants. Right here, if I reduce all the upper canopy, take all the upper canopy leaves, I go from this to this. These are my sinks. These are my sources. And when I remove that source, I get a small sink. And our growers make their money by making big sources and big sinks. And so what that does for us in the genomic realm is when we can look at what our products are doing and everything that goes into that source to sink relationship, sunlight, photosynthetic activity, carbohydrates, translocation of carbohydrates, certain nutrients, that allows us to really address this source to sink relationship. I worked my way through college for Dr. Tom Ramage. He was a barley breeder. And what he drilled into my head is the phenotype is expression of your genotype within an environment. And the genotype is simply my genetic constitution, all the genes that go into making a corn plant or a potato plant or a strawberry plant. The phenotype is the observable characteristics of the individual interaction with its genotype and the environment. This is what I physically see when I'm out walking in a field. He also drilled into my head this thing called phenotypic plasticity. And that's really the ability of a plant to change to stimuli or inputs from the environment. And sometimes we forget, Ben and me and any other agronomist are part of the environment. We're making decisions to put things on our plants that are either going to be or should be stimulus. And our job actually every day as an agronomist is to maximize the phenotypic plasticity of that corn plant in whatever environment it's growing in. What can I do to make it yield more when I got a rainfall? How do I help it handle 5 days of high temperature? What Layne Ellen just explained to us opens up this possibility really, really well. And we'll walk through that. You'll hear phenotypic plasticity over and over and over again. So in these enriched pathways that Layne Ellen was explaining to us, biostimulants and crop nutrition are dependently linked. You can't separate them. You separate them, it's never good. You have to understand that if I'm going to put a biostimulant out there, there are going to be a group of nutritionals that need to be present in the plant, or you have to adjust for that biostimulant to work at its highest possibility. And the only way that we can ever learn that and use it effectively and use it to where we're helping our growers use biostimulants at 95 to 100% positivity with return on investment is to understand this nutritional by biostimulant interconnection. And that's what really this genomic work has really allowed us to do very, very well. Because now I can look under that surface of that water. I'm not working in the dark anymore. I know where that sandbar is, so I can run my big high yielding yacht of barley fields at a pretty high speed and learn how to navigate around that sandbar. It's a must to understand. So making biostimulant works. Biostimulants act at the gene expression level, signaling or constructing compounds and pathways that have a gene expression requirement. The products or actions of the above are always, not sometimes, not maybe, they always have a nutrient dependency. The key to biostimulant performance is understanding numbers 1 and number 2 above and making sure that the dependent nutrients are addressed before the biostimulant is applied. So what I want to talk to you, what I want to kind of put it into real life out here is I want to do a little bit more on the time and durability. Layne Ellen did that really well. And then I'm going to walk through 2 very relevant scenarios on this using genomic data to build the correct biostimulant, put it on at the right time, make sure the complementary nutrients are with it. I want to walk through two very relevant scenarios, an abiotic stress mitigation scenario and a nitrogen use efficiency scenario. So timing and durability. Layne Ellen covered it very, very well. But from an agronomist point of view, being able to understand what genes that I'm having a positive effect on and what they actually do allow me to basically create, through phenotypic plasticity, I can create the most optimized genetic plant for the environment that I'm growing in that given year. So the first thing we always look at that it really allows us to do, well, this genomics where it really allows us to do is really pinpoint this very, very well. The very first biostimulant biological I ever used was in 1983 on my father-in-law's cotton farm in southeastern Arizona. Guy bring us some biology to take care of sodium, or we had high sodium soils. And the first year it worked really well. Second year it really didn't work that well at all. Third year it didn't work at all. And what we learned was live organisms die. And at that time we didn't know how to keep them alive. Fast forward about 5 years and a company comes out with a cytokine to put on cotton, and I'm consulting in West Tennessee at this time. And they had kind of educated us on cytokines, but they didn't educate us really well. And if they would have had genomics behind what cytokines were doing and dependent nutrients, one part of my life would have been a lot happier. But we put it on the first year, had good rainfall. Our bowls were bigger, made anywhere from about 200 to 300 pounds more lint cotton to the acre. One field made a half a bale, or a whole bale, 500 pounds more to the acre of cotton. Then next year, we did the same thing. And quite honestly, our yields went backwards and our cotton turned red on us toward the end of the year, which is never a good thing if you know anything about cotton. We began to scratch your head saying, well, the only thing we did different was put this cytokine product on it. What we learned there, we got a little dry, and we learned that cytokines were potassium dependent, and we went potassium deficient, and we simply couldn't feed our plant. We had bigger yield potential, but we simply ran out of potassium. That was a great learning time for me. So understanding rates, applications, if I would have known what I know genomically now, that field would have had a lot of potassium. It was kind of at that time, we were sitting on the top of the lake, having no idea what was underneath us. When we look at when we should apply, let's say we have a product that that is really going to increase certain nutrient uptake. Really going to help me in stress management. I'm probably going to want to try to apply it out here. My durability then tells me how long is it going to last. So let's say this product here that I put on right here, let's say that from what we've seen in our genomic studies, because normally what we do, what Layne Ellen does for us, is we look at a 3-day after application, a 14-day application on corn, and then VT or tassel stage. So we look at all this enriched pathways at these 3 points. If I've got a product that has an enriched pathway and I'm still showing very good upregulation of the genes for stress and energy production, my durability means that I don't have to spray again during the whole year. If that same data shows, well, you know what, by the 14th day, you're kind of beginning to get lost and some of the stuff is beginning to fall out, you may decide I need to spray again right here. So what the genomics have really done for us with our biostimulants is hone in the correct time for what this product is actually doing to the plant. If this is a nitrogen use efficiency product, or it's going to convert nitrates to proteins, and the proteins is going to make money, I want to put that on right here because my nitrogen uptake curve goes like that. I surely don't want to be putting on a product that gives me nitrate conversion out here. My plant's either already suffering, can't make proteins anymore, is already struggling with other things. So again, one of the most important things we see with understanding the genomics is when do I apply it and how long is it going to last and when, if any time, do I need to come in with another one? Take this nitrate conversation and take it to your wheat, right? I want to put it on. If this was a wheat field, I'd have almost the same nitrogen uptake curve. So I'd want to get it on early before this happens. But if I need high protein wheat, I'm going to come back out here at Flagleaf and reapply a product that can do this, because I can at this time positively affect protein content in my grain. Again, understanding the genomics and understanding how long that genomics lasts, the durability, really hones in my timing to where we can go out there very confidently and get 90 to 100% positive rates year in and year out. For two reasons. When we put it out there, it works. For the second reason is if we're out of whack when the guy wants to do this, we simply walk away from the cell and say, we'll come back and fix it next year. So it really, really helps us recommend the right product at the right time, and if and when I need to come back and do it again. So let's talk a little bit about abiotic stresses in a scenario and a biostimulant what should a biostimulant look like that's going to control abiotic stresses, and what is the durability behind it, and what nutrients are important to go along with that biostimulant. So I'm going to walk through some work that Dr. Ismail Çakmak out of Sabancı University in Turkey is doing. I've known Ismail for a long, long time. About 20 years ago, he educated me on the nutritional needs for the physiology of germination and emergence. He's probably the best crop nutritionist in the world, and I want to give him credit for this because these are slides he's provided to me. That's kind of the key where his research on him. The key player in stress management, especially drought or low water use, it doesn't have to be drought, just but limited water use and heat, is the creation of abscisic acid. Abscisic acid actually drives the closure of stomates to conserve water. There's a lot of biostimulants out there that have been reported to produce ABA under stress conditions, but a lot of times they still don't work, or they don't last long, or they're limited in their effecture. So knowing this, that abscisic acid is important to water stress and heat stress management, we can start thinking about, okay, does my biostimulant have a influence on how the plant utilizes and moves abscisic acid? But the story is a little deeper then too, because it's not that easy. We know that when we get water stressed, abscisic acid concentration rises. That was some work done in 2002. So we know that it has a role there. This was in corn. We also know that sulfate is the only ion that shows consistently higher concentrations in the xylem sap during drought stress compared to no stress. So what I mean by that is we don't see the plant taking up more nitrogen when I get water limited. We don't see it take up more phosphorus. We don't see it take up more potassium. We don't see it take up any more calcium. Of all the ions looked at in this study, sulfate was the only ion showing consistent higher concentration. So from a practical agronomist, physiologist, I got to look at this and go, I wonder if there's a sulfate role in drought stress and limited water stress. So in 2022, Dr. Lee did some work, and lo and behold, he looked at time of stress, 0 to 15 days, ABA concentration, and sulfate concentration. And here's my concentration graphs. So you've got to look at this and say, that's highly coincidental, or it's highly correlative. And so at the end of the day, you got to say, well, I really don't think it's a coincidence. And so you either got to say either this drives this, or this drives this. And that becomes real important which way that goes. So Dr. Çakmak has been studying that. And here's what we found out. Sulfates turn to sulfides in the plant. They turn to cysteine, which is an amino acid. To make, there's a protein in here, protein/enzyme in here, that is cysteine required. It's cysteine dependent to make abscisic acid. There's not enough cysteine to go around. If I'm not converting my nitrates, and we'll talk about that in a minute, to amino acids, and I don't have enough this happening, I can't make cysteine. If I'm deficient on sulfur, I can't make cysteine. And if I start getting drought, and this is limiting, this is a source to make a sink. If I can't make enough of this, guess what? I don't make enough ABA, and I got a plant that can't handle the stress. So this was actually old research where we found this, but that's a really interesting relationship. So let's say I have a biostimulant that helps in the ABA pathways, but I don't understand this. My biostimulant is probably very inconsistent in its work. So, importance of understanding the genomics of stress, timing and duration become really important. When am I going to put that product at? How long is it going to last? Pairing up with dependent nutrients becomes huge. Sulfate is now, we always used to think potassium was my most important water stress nutrient. New knowledge, right? Keep being a student of the game. New knowledge tells us we better be looking at our sulfate. If I want to start screening my plant to understand what does it really do, and I find out that my product actually increases sulfur uptake, actually helps in the cysteine, the cysteine processes, actually helps in the conversion of the sulfate once it gets in the plant to sulfide, where the plant can use it. But at the same time, it actually plays a role in promoting or optimizing ABA content. It increases photosynthetic activity. All those things that I learned from a genomics study tells me, you know what? This should be a really, really good stress mitigator. And then if I understand those pathways, I can look up those pathways and begin to say, well, is it sulfate dependent? What other amino acids need to be made here? And I can begin to pair up my biostimulant with my nutrient. This also allows us to be proactive, not reactive. I can create this type of plant if I understand my genomics, my biostimulant, and my nutrition. That's phenotypic plasticity, right? I'm not reacting to when a stress is coming. I'm creating by being proactive. I can create a plant that's going to handle the stress much better. This is what Scion does. This was work in the north central part of the US, in North Dakota. It's spring wheat. Understanding our product, working with Layne Ellen, fully understanding what our product does, we knew that the correct timing for Scion was as early as possible. The first trip across that field that a farmer is going to make, I want Scion in the tank. Because what we know Scion does, it creates a plant that wants to create. And it creates a plant that can actually be, that phenotypic plasticity is I'm creating a plant that knows how to manage that abiotic stress much better than the plant that doesn't. Same genetics. This is my untreated check. This is Scion. It was sprayed at 22 ounces at the 3 leaf stage. Same genetics, same planting. There's the difference. There's the treated part. There's the untreated part. Okay. Notice the durability of Scion. This is early on, early tillering. This is reproductive. Look at the difference in the phenotype of this plant. We changed the phenotypic characteristic of that plant within this environment. This barely in boot stage. This pollinating. Right. I don't know how many people use degree days in their work. But what I've always told, and this was taught to me by Dr. Stith, is that a heat unit is not a heat unit when the plant is stressed. And that really shows us. Same sunlight, same degree days. Look at the difference in how this genetics, how this same plant, same genetics, just optimizing the genetics, is handling that season versus how this one is. Right? So let's move to nitrogen use efficiency scenario. Same principles in mind. How can we turn every pound of applied nitrogen into more yield? How do we get nitrogen use efficiency? This has an economic return to the grower. It's got a huge environmental impact. So keep in mind, nitrates are plant available. They are not plant usable. The plant can't use nitrates. Takes them up all day long. Plant likes nitrates. The plant's got to do a lot of things with nitrates before it can ever turn into return on investment for our growers. So let's talk a little bit about nitrogen uptake. Nitrogen uptake is controlled by proteins. You have what we call HATs and LATs. These are high affinity transport systems, which is protein system, and low affinity transport systems, again, proteins, and roots. That's what drives uptake. My high affinity transport system is when my nitrate levels are low in my soil. My low affinity transport system is when my nitrate levels are high in my system. As you can see over here, plants have HATs and LATs for nitrates, only HATs for ammonium. Amino acids, still not quite characterized, but even urea, a lot of people don't even know it. The plant will take urea up. It actually has urease genes, most plants do. We just found that out in about 2010. But you notice for urea uptake, it's all about high affinity transport systems. So again, by understanding the genomics, I can look to see if my biostimulant actually has any impact on these protein productions and how long does it have an impact. And can it, when nitrate levels are low, can my biostimulant help the plant create more of these? But then when I put a side dress application on them, can my biostimulant help me create more of these? I'm never going to know that if I'm not looking at enriched pathways, not just that heat map, not just the up and down regulation that Layne Ellen talked about, but the very enriched pathway she talked about. So again, a little bit about HAT and LATs. The plant wants to express these early vegetative. Once I get to reproductive, the expression of these genes goes down. Both in this is a LAT, this is a HAT. Both of them, by the time I hit reproduction, it goes down. This knowledge, along with the work that Layne Ellen does, allows us to time a biostimulant correctly if it has an impact on these genes. So that's uptake. What happens to get the mobility in the plant? Nitrate mobility is 100% potassium dependent. In the leaf, the plant makes potassium malate. And so in my leaves, the plant makes potassium malate. It moves it down through the phloem. It sends the malic acid out to the root. The potassium turns back around, picks up a nitrate, carries it back up to the leaf to where now I can utilize, now I can put it in the leaf to take that nitrate and turn it into amino acids and then turn it into proteins. Okay. So a biostimulant that helps me get potassium into the plant is going to give me much more nitrogen use efficiency than a biostimulant that has no impact on potassium uptake. And this is going to be more important in my very sandy soils, in my heavy clays, where K uptake can be limiting. So again, the only way I'm going to know this with my biostimulant is to look at the pathways of potassium uptake. So now I'm in my leaf, right? Still can't make any money. My nitrate is still unusable. Okay. I've got to make two enzymes. I mean nitrate reductase. I got to make nitrite reductase. To make this reductase, to make this nitrate reductase, which takes one oxygen off, I have to have potassium, I have to have sulfur, I have to have iron, I have to have moly, and I got to have high sucrose. So this means high photosynthesis activity. So now a few other nutrients come in place. Nitrogen, manganese, iron, other nutrients involved. Highly photosynthetic machine, potassium, sulfur, iron, and moly to make this enzyme. I knock an oxygen off, still haven't made money from my nitrate. I'm at nitrate. I've got to make this enzyme. That's a sulfur and iron dependent enzyme. I knock two oxygens off. I go to nitrogen. The plant makes ammonium, actually very easy. I can almost make amino acids and proteins. Can't make them yet. I'm getting really close. So in the plant, I've got to make, it's debatable. Somebody will say 20, some say 21 essential amino acids. Okay. To make these amino acids, I have to make either glutamate or glutamine first. All amino acids come out of the production of these two. If I can't make these two, I can't make the other 21. I can't make proteins, and my yield will go down. So our product called Uplink, there's a little metabolite that runs this organism, this glutamate-glutamine synthase process. There's a metabolite that's the linchpin. It's the key to making this run right. That's what's in Uplink. So I get my genes up here running right. I get nitrate into my leaf. I'm getting this reductase running right? I get NH4. The faster I can do this, the more efficiently I can do this, the more amino acids and more proteins that I make. Now realize to make amino acids and proteins, I need hydrogens and oxygens and carbons. So I got to bring CO2 in. I've got to bring water in to get to my hydrogens to make this. That's working here. But again, if I can look at the genomics, the enriched pathways, I can look at what does my biostimulant actually do and what nutrients do I need to put it with. So let's look at a little genomic story behind Uplink. We know that it has a positive impact on the hats and the lats. We enrich those. We know that it enriches nitrate reductase and nitrite reductase genes. So now we have uptake here. We have assimilation there. This is the key, right? This is the main pathway that it does really, really well. Layne Ellen also showed us that it has a positive impact on Rubisco. So when that CO2 comes into my plant, I need Rubisco to utilize the CO2. It also goes past just the carbon fixation or utilization. It actually helps make a lot of carbon metallogens. So I'm making more sugars, I'm making more starches, I'm converting these to energy. And there's some other domains there. So going back to our durability conversation, and understand what this product does, what Uplink does, and how long it lasts, I know that I want to, number one, put it on as close to those high nitrogen demands as I can. I also want to make sure that I'm pairing it up, or I know that my K, my S, and my iron, and my moly are in good shape, and I'm under high photosynthetic activity, right? Or I could put Uplink on and have minimal positive effects if my potassium is deficient. So again, understanding this relationship with the biostimulant, understanding the genomics, and the pathways we enrich, I can look at those pathways, go back and understand what nutrients help drive that pathway, pair this up, and my positive results are just much better. It too is proactive. Part of the proactive is making sure my nutrients are right. It too has what I call phenotypic plasticity. I'm creating more inefficient roots. I create the nitrate transport system. I make amino acids into proteins. And I can do that when the plant is in the early stages and have a totally different plant at the end of that because of this phenotypic plasticity. This is actually a trial in Wisconsin, United States. This right here is 170 pounds of nitrogen with uplink. This way right here is 230 pounds of nitrogen without. This still had good potassium, had good sulfur, had good iron, had good moly because we put it into our protocol. This did too. Same fertility program, just difference in nitrogen. This is nitrogen use efficiency. This is taking nitrates, turn them into proteins, bringing more CO2 in. Look at the biomass difference there. I've got a much better source here than here. My sinks, my yield is going to be much higher here than here. So I'm coupling what Dr. Stith told me about never ever forget the source of sink relationship. I'm bolting that on to what Dr. Ramage drilled in my head, that my phenotype is expression of my genotype within the environment. And Tim, an agronomist, a good agronomist, knows how to make phenotypic plasticity within any environment that he's planting his genetics in. And that's what we're seeing here. So summing it up, having this genomics knowledge provides clear vision and insights into phenotypic plasticity that is provided by a nutrient and or biostimulant. We can get the correct time to apply the product. We know the correct duration and need for multiple applications. We have high ROI and success rate. And at the end of the day, the question becomes, are you in the dark or on the top of the lake, or are you going to go to the bottom of the lake? And with that, I will turn it back to you, Ben.
The way we're feeling in the UK, at the moment, I feel very much at the bottom of the lake. We've had a bit of a wet time. Nice to hear that you're working on drought tolerance. Just wonder whether you're working on anything that survives under 3 foot of water. Wheat in particular would be quite useful at the moment. I'd be interested to hear your comments on anything but drought tolerance. It's been a rough winter.
Yeah. Well, kind of on that we have. So if you look at some of the things you want to do to get a plant out of that water stress, one of the things you want to do, so when that plant gets under any stress, especially water stress, your ethylene production tends to shoot up because it's telling the plant to stop growing, right? And when that happens, we get what we call deliproteins made, and deliproteins tell the plant stop, there are breaks. So any biostimulant that will kind of stop that ethylene conversation. So as soon as you got, as soon as you can kind of come out of, you get the water off that ground, the air kind of moves back into the soil, things can grow again. To get that thing growing fast is to shut down that whole system as quick as possible. And there are actually there are biostimulants that actually do that really, really well. One of the things we did see in Scion is you actually do have control of monitoring that ethylene production in a stress. You can keep it at bay, I guess would be the best thing, or mitigate its response.
Excellent. Just a quick one for those putting questions in the chat. any chance they can copy and paste them into the Q&A? It just makes it a bit easier to manage the screen. And without further ado, we've got quite a few in the Q&A. I see Layne Ellen has answered the one question. I don't know whether you want to just elaborate for the rest of us on the question and answer, or do you want me to answer it, or ask the question? Or yeah. Okay, cool.
I think if I understood it correctly, the question was about if we see this, because biostimulants are kind of notorious, or biological products in general are notorious for this variability that we see of their effects in the field. Do you see that same kind of variability reflected in the gene expression? And I would say that yes, to some degree, but this is where it's really important to look at entire pathways and to do what we would call like characterizing the responses. The degree and intensity of the response that we see might vary a lot, but often we'll see it's the same kinds of responses. It's just other conditions and other impacting factors can kind of vary how much of that response we actually see. So it's kind of like if you eat something that's salty, it's still salty. It still tastes like chicken, but it might be more or less salty. Like it's still the same type of response often, but it'll just can vary in its intensity.
Excellent. So I'll just go down the questions in order. Trevor Smith has asked, can you give us the best example that you have experience of biostimulant activity that's created the largest uprating of gene expression and therefore plant effect? I presume that could be in the lab or in the field.
Can you repeat the question? I'm not sure I quite understood it.
Well, I don't understand it either. I mean, Trevor's just talking nonsense, but we'll have another go. Can you give the best example that you have experienced of biostimulant activity that has created the largest uprating of gene expression, therefore plant effect? In other words, what have you seen where you've put a biostim on and seen the greatest plant effect from?
Yeah, well, I think there's probably two examples in that. So there is one biostimulant that we look at. Well, the nitrogen use efficiency one, I'll go back to that one. On that one, in given fields, we've actually been able to see our bushels created per pound of applied nitrogen go up really very dramatically. In some fields, again, in some fields that tend to lose nitrogen, so we're getting in the plant quicker. We've seen 17, 18, 19% better yields and better nitrogen use efficiency. Another example, I think, is in West Texas, where we're growing corn and wheatledge, corn silage and wheatledge silage for dairy cattle under very limited irrigation. It used to be high irrigation. We used to have all the water we wanted. Now we have half the water we need. And a lot of that water has become very sodic, a lot of sodium in it. We've actually been able to look at changing our nutrition program, getting the right nutrients out, and a biostimulant out there, and almost maintaining our same yields as we have with full water, and we're actually seeing an increase in overall crude protein. So those are the agronomic characters that really makes you understand what biostimulants can do when you're under those certain type of conditions. What we did in both of those cases, we had the proper nutrition. We understood what we're trying to do, so we lined our nutrition up and put the biostimulant in the environment that's going to do what it's supposed to do.
I also have an example if you're interested, if you want to hear it.
Yeah.
I was working with a client before that had a product that was really helpful at, it's drought stress, but it was helping a lot with drought stress, heat stress. And what they found was they put it in, this was in corn, usually around V7 or so kind of mid vegetative growth, early to mid, and had great responses. Well, one year they were late. It rained and rained and rained, and they were late getting it out. And the plant was already up to about V12 and starting to tassel. It was really close to reproduction. And when they did that, their yields tanked. They were like, okay. And so we did some work and actually looked at the response was really tricking the plant into getting it into thinking it was being stressed. And so it was causing it to spike this abscisic acid and the ethylene response that Tim was talking about, which when they did that early, it didn't seem to really, the plant was able to kind of recover from that pretty quickly. And it was still really focused on vegetative growth. But when they waited until reproduction, it punched it so hard with this ABA and ethylene response that it actually kind of shut down that maturation and negatively affected their yields. And so for that, it wasn't until we could understand, it wasn't that they were getting improvements in yields from the years before because of a growth stimulating response. It was because it was helping it to mitigate stress. And so being able to differentiate between promoting growth versus allowing the plant to grow despite stress gave them that answer of why a late application was actually detrimental for them.
Yeah. I mean, from my own experience using Scion, in actual fact, we used it for years on wheat, and we're seeing some interesting results. But the year we put it on faba bean was the time where we just saw it totally do something that was just beat every fungicide and any other choice of anything else we'd seen, and that was the revelation for me seeing that, and it kind of leads me on to a bit of my own question, which is, are all these biostimulants crop specific? Because are these just one trick ponies that will? It doesn't matter whether you've got potatoes, tomatoes, wheat, or rye, or should we be looking at different doses on different crops? I often see dose rates are often at the value of the crop. So you often see far more. You're allowed to use 2 or 3 liters on a crop of potatoes, but only half a liter on a crop of wheat. And is that anything to do with the crop, or is that just what the price will stand?
It's a great question. So a few things, from a physiology point of view, you can, if you understand what we talked about today, you can look at a biostimulant you have and say, okay, it worked this way in corn. We've got genomics data on cotton with Scion. It worked this way in corn. We know what it's doing physiologically. We know the nutrients that it takes here. I ought to be able to connect a fairly simple dot to go to another crop to look at where I want to do it, right? Layne Ellen, there's a lot of things within crops on some of these genomics. Layne can explain it better than me, but we call them conserved genes. It's really basically the same genes that are doing some things, the things in photosynthetic activity or stresses in a cotton plant that's probably being done in a corn plant. So we can make those logical conclusions. Timing becomes a bit different when you start looking at, so what am I trying to accomplish here, right? So in a cotton plant, I'm trying to accomplish some other things than I am in a corn plant or a wheat plant. So then it comes down to the timing and the durability. So uplink is pretty much, you can take that product across any crop because it's that glutamate synthase process for utilizing nitrates and turning amino acid proteins is basically the same in all crops. Soybeans, even though the legumes still use that process. Once I've got the fixed nitrogen to ammonium, it still uses pretty much the same process to get the amino acids, still goes through that process. So that's what helps us. Again, that's why it's so important not to be at the bottom of the lake and not to be in the dark and understand that is you now learn how to make that product work in other crops. One thing we have learned that I think is interesting, Ben, in cotton, having looked at the genomics work, Layne Ellen and I one time said, really knowing what Scion does, if I went 10 ounces, 10 ounces, 10 ounces 3 times at all the times that the farmer crosses his field at key physiological growth times, it ought to work better. So we did that on a proof of concept last year, and sure enough, our standard rate of how we do it actually out yielded it. So again, understanding the genomics and understanding what it does and understanding kind of the durability allowed us to maybe make the product work better in certain crops.
I think it also depends on if you're talking about a product that elicits plant defense responses for a disease resistance kind of result. a lot of the efficacy there is also going to depend on the life cycle of the pathogen that is giving you the disease pressure. And so that timing might look very different for you all versus, growing wheat versus somebody growing wheat here, depending on, number one, the types of pathogens that are giving you the heaviest pressure, but also the timing when conditions are most conducive for that disease pressure. And so if you're using something that elicits defense responses that makes the plant more resistant to that pathogen or that disease, then the timing of when you need to prime that plant for that might be earlier for you than it would be for us.
And to add on to Layne Ellen's, number one, she's an expert at that. She did her PhD in this whole thing, and she's pretty bright in it. So I'll pat her back on that one. And that's actually, that biostimulant is actually, probably could go back to your earlier question, was one of the most dramatic things once you learned what it was doing. But to her point on plant disease, I think there's a question on nutrient needs. Yes, you look at the product that she looked at in her PhD, and you look at the pathways it's doing. There's definitely a role for calcium nutrition in that pathway. It actually has some positive effects on making what we call our SODs, or superoxide dismutatases, work better in plant diseases, and those are hugely copper, zinc, and manganese dependent to make those. So I'll stand on this until somebody proves me wrong, and I'll buy them the most expensive Wagyu steak that I can find, not the one you can find, but I will be fair. And I don't think there's any time that a biostimulant will not have a nutrient needed to make it work at its best. May it work all right? Yeah. Will it work at its best? No. And the more you understand this nutrient to biostimulant by genetic rule, your biostimulants are just going to work better. So I think it was Trevor's question, and I hope I answered it for him.
Excellent. It does look like we've run out a bit of time, and I think that's answered Andrew's question. Just the very quick last one from Andrew Howard saying, I think you mentioned it, and I think he missed it. So, what is the nutrient dependent of scion? And then we'll wrap up.
Well, nutrient dependency on scion could be really what I'm going after. Scion is going to help me with my nitrogen use efficiency. So my potassium, sulfur, and iron, and moly, because it's got one of the metabolites in there is a metabolite very closely to what's an uplink. So I get better nitrogen use efficiency for those. On my drought, if I'm going to try to mitigate, I'm worried about, I want to build a plant that's going to handle limited water and some heat better. Then I'm really going to look at my sulfate, my sulfur nutrition. And it's got to be sulfate. Remember, sulfate's what the plant takes up. So I want to look at using a sulfate form of fertilizer, and I still want to look at my potassium. So you look at scion, and if I get my sulfate right for my mitigating my heat, my water, I've also covered it to make proteins, helping me reduce my nitrate. So it's kind of twofold there.
Excellent. Right, I think we best wrap up, and left for me to thank Tim and Layne Ellen Harris for an excellent presentation. I think we've got a bit of a poll coming up. I think it's already on the screen, but also thanks very much for BOFIN and John and Andrew Crummy as well. So we're not allowed to vote, but if you can, that would be very much appreciated.
Just want to say thank you very much, Ben, for chairing that so ably, and also to John for supporting the webinar, and of course to Tim and to Layne. But I mean, that was excellent. I tell you what, that is just mind-boggling. We will distribute the recording of this, and that will have all the slides on it. There is a request, by the way, Tim and Layne. I don't know how you feel about this, for your slides themselves. I mean, I guess you can look at them on the video, which we'll definitely distribute. But there's been a request for the slides, so if you can share those, that would be great, and we can send them to anyone who wants them. There are loads of questions that we never got round to. I tell you what, I'm going to be watching the video again when it comes through, because there's just so much information on there. It's just blown my mind. It really has. There's so much to go at here. It's brilliant. So thank you so much to everyone who took part. Thank you very much to all of you who put in questions. We will try and get answers to those questions, but we'll try as hard as we can. But there are an awful lot of questions that I'm afraid we never got around to. So I apologize for that.
Yeah, Thomas, you can populate the questions, and we have the people who answered them. Ask them, send them off to Layne and I, and we'll be happy to answer them.
Oh, that's very kind, Tim. No, thank you very much for that. Well, look, let's wrap it up now. But thank you so much, all of you, to Ben and to Tim and to Layne and to John and to Andrew as well for being here as well and for answering the questions. And thank you to all of you for joining the webinar. This has been a record webinar, by the way. We've had masses. We've had over 300 people who registered for this webinar. So it's been a real success. And as I say, I'm looking forward to receiving the recording. So I'm going to call a halt to proceedings and say goodbye to everyone.
Thank you.